again. So the next topic we're dealing with is using tagging data. Sorry, it's a little bit out of order, but probably good for the people who were here for the spatial modeling workshops. And so first off, Dan's going to give us the keynote, and then Rich is going to give us a comment after that.
All right. Yeah, I'll try to make this quick so you guys can all argue about tag mixing for a while. So yeah, I just want to note my co-author here, Matt Vincent, who it was really good to interact with and have on this in this keynote because he actually has some applied experience. When they asked me to give this presentation, I was kind of going to use my background and what I'm used to talking about in terms of tag integrated models. And Simon and Mark both told me, no, that's not how you can approach it for tunas. And I said, all right. And I swear that's not exactly how I feel, but maybe a little bit. But yeah, so it was good to have Matt on board since he has some actual applied experience with tuna tag models, whereas I'm more of a groundfish coastal species kind of modeler. So my experience is really in these kind of small management areas like New England, whereas I know you guys all are used to these giant kind of ocean basin situations. But I do think that kind of your background may have a bit of influence on your perspective in terms of maybe the usefulness or the need for tag integrated models. So I think it is good to have kind of this mixture of experience between the two of us. At the same time, anyways, yeah, so I think it also creates new options if you're looking at different species. So on the left here is sablefish with a satellite tag. That's a species I kind of work on now. And as you can see, the satellite tag is about as big as the fish. So it has some strong influence on whether that's actually useful information, whereas on the right, it's kind of a satellite tag on a tuna. So, the proportional size of the tag is much different. So you have, new options when you're talking about these different types of species like this. So there's some pluses and minuses, I think. I've given this presentation to CAPAM, I think 4 times now. So I swear I didn't just copy and paste it, but maybe I did a little bit. But it is kind of interesting to see the progression of how kind of talking about spatial models and tag integrated models has changed kind of over time here. And it is nice when I give sort of these presentations to more coastal groups, where I usually interact, everyone goes, spatial models, not that interested. But I think here, talking to tuna people, it's nice to have a bit of a different perspective on the usefulness or the need for these kind of spatial and tag integrated models. And it's also the fact that, this is the abstract from that special issue for the 2018 CAPAM Spatial Stock Assessment meeting. And it's really focused on the fact that there's not a lot of spatial models, but the real caveat to that is that, obviously you all are used to doing spatial models, so it's much more popular to use them, or maybe not popular is the right term, necessary to use them in the tuna world. So yeah, it's nice to have kind of a different perspective. So I guess the question is, this is a slide on the right that I used for 2018, where I kind of emphasize don't overthink the use of tagging data. And the question is, does that differ for, when you're talking more explicitly about tuna? I think generally speaking, it doesn't change the advice, but it does make things a lot more complex, I think. It's the same issues, it's just a different kind of scale of those issues. Again, this is a picture from one of Matt's assessments. I don't even know what species it is, I'm sorry. But it does kind of illustrate the point that you're dealing with these giant ocean basins and dealing with mixing of fish across these huge regions is a huge issue. So I think the main point here is that it changes from don't overthink it to think about it. And that doesn't really help probably in the conversation here because everyone already knows that, but it's good to emphasize. So I'm gonna do this as quick as possible. I'm gonna try not to talk too much. I think everyone kind of has the background here about what tag integrated models are, what tagging can kind of influence and how you can use it in assessments. So I'll quickly go over them, introduce some of the main issues, give some advice, I guess. I don't know how useful it is in the tuna world, but I'll try to introduce some things that me and Matt talked about. I'll go over some slides that Matt put together for how tagging data is used in the tuna world and just some future directions that we kind of discussed. So This is just basic, there's multiple kinds of tagging data. I think the one that we're going to talk about the most today is conventional tagging. And there's obviously pros and cons to all these different tagging types. For conventional tagging, it's generally the relatively low cost. You can put a lot of tags out for a lot less cost compared to electronic tags. But you're left with a lot of low resolution data. Everything's pretty much unobservable states except for the release and recapture locations. And then you have all of the nuisance parameters, which is basically what the title of the talk is about. Conversely, you have, sorry, all the formatting got messed up on this, different versions of PowerPoint on these computers. Anyways, so yeah, there's electronic tags, and with electronic tags, you got the archival, satellite, acoustic tags, all sorts of different kinds. You get much more high resolution data, but there's always questions about representativeness and different kind of representativeness, because now you only have a few tag tracks. You know exactly where those fish went, more or less, but you can't put out 100,000 tags. And then there's also the use of natural tags, which I think is kind of an interesting intermediary between the two. So it's kind of a moderate cost because generally speaking, you don't have to put tags out, they're already there. All you have to do is go out and collect them. But really the problem here is you need to know a lot about the life cycle in order to interpret what those tags mean when you're talking about either otoliths or even parasite infestation. And I always like to cite that figure down on the lower right there is from Karen de Moore's paper in South Africa, where they used, I think it was sardines, but parasite infestation for sardines to estimate movement between two regions off of South Africa. And I think that's a really cool use of this kind of parasite data. And then also, I think this is becoming a lot more popular, and there's a lot of interest in this, is using genetic tags and close kin marker capture. And obviously it removes some of the nuisance parameters that we see in the conventional tags, but there's other issues involved, including cost and extensive sampling effort, things like that. So again, Simon and Mark kind of asked me to focus on conventional tagging, so that's what I'm going to do, whether anyone wants me to or not. So yeah, what can you learn from tagging data? Obviously, there's a lot that you can learn just by looking at the tagging data external to an assessment. It gives you information on migrations, population structure, things like that. You can do it, obviously we talked earlier today that you can do external estimation, use that to estimate parameters outside of an integrated assessment. But you do lose some of the information content, and you have some issues about, Matt Vincent already mentioned earlier today that you have some issues there. If you fix parameters in your external analysis, you lose some of the information content. You have issues with carrying over uncertainty and things like that. So yeah, today I'm going to focus on the tag integrated aspect, so incorporating the tagging data directly into the assessment. So essentially you're simulating or it's not simulating, you have a tagging submodel, so you you have the number of releases by area, things like that. You put those into your model, you model them simultaneous with your untagged population, you have combined objective functions, and you use that to estimate different parameters that are shared across tagged and untagged populations in your assessment. There's a lot of ways to go about modeling tagging data. I'm not going to get into them too in depth, but we talked about earlier in our spatial modeling workshop, I think Castle uses more of a Peterson estimator as a way to estimate more of the abundance by area if you use the tagging data. And a lot of other models, and I think mostly in the tuna world, is kind of this tag attrition approach, where you're kind of focusing on estimating mortality and movement. So yeah, I'm going to focus on those. And so what are the issues with using tagging data? Again, I think everyone here is probably well aware of all these problems, so I'm not going to harp on them too deeply, but I think the number one issue is incomplete mixing of tagged fish. So you release the fish, it takes a certain amount of time for them to mix across the entire region that you're modeling. Sometimes they don't mix at all, they don't fully mix ever. And so, trying to deal with the bias that induces in many of your parameters is a huge issue for assessments. And then you have sort of these field issues, which is what happens, how do you deal with, when your tag fails, when you have mortality of a tagged fish, the tag falls off, tag loss, things like that. And then there's also trying to get tags back. So when you have misreporting of information or just non-reporting of the tag itself. And all of these things, if not accounted for properly, can lead to bias in your estimates. That figure in the middle is one that I did for yellowtail flounder. And that's just a simulation estimation model, and basically showed that if you fix the reporting rate at the wrong value, then your estimates of SSB, F, and everything become highly biased. So, there's lots of issues when you start getting bias in your tag, what I like to call the nuisance parameters. And then there's, the issues of sampling, getting spatial temporal coverage that covers all the regions, all the age and length classes, things like that, and just making sure that the dynamics of the fish you're tagging reflect that of the entire population. That's one of the issues kind of with the electronic tags, is if you only put 2 or 3 tags out, does that really represent the whole population and how they move? And then there's also the issue of data conflicts. So yeah, that's a big one in terms of data weighting. You have this tagging data, you put it into your model. How do you kind of downweight or upweight your tagging data there? And then of course, when you already have that in kind of a spatial context, it becomes very complicated, as Mark pointed out about an hour ago, 2 hours ago, maybe 3. So, I mean, how can we deal with some of these nuisance parameters that I talked about? The big one, again, is incomplete mixing. And I don't think any of these have a solution per se. I think there's ways to handle them generally. Whether it's a perfect way and if it reduces the bias completely, I think is still debatable. But I think you can start addressing some of these issues. And this paper that Simon was a part of, I think, is a good one that you can kind of start analyzing your tagging data to look at whether your fish are actually mixing. And I think this one was, this figure up here on the right was looking at the density of tag returns from a release location in comparison to boundaries, or regional boundaries within the assessment. And basically looking at whether you have mixing within those boundaries and whether you have mixing outside of them with the other regions across the assessment model. And I think that one was called TART. I don't know, it may have been custard, I can't remember. But yeah, so in terms of dealing with incomplete mixing, I think there are 2 main ways that are normally used. One is kind of censoring the data, so taking out data during the first couple periods after release for fish that haven't fully mixed across the region. And then the other one is kind of a similar approach, but instead of taking them out of the model, just estimating different parameters. So you could estimate a different F during the mixing period. I think you can also try to estimate a different movement rate if you think that the tagging process has influenced how the fish are behaving after they've been tagged. In terms of kind of these field issues, some of these recommendations were made from my personal experience that we've used for things like yellowtail flounder, which are very different from tuna. Simon kind of pointed out that they were kind of dumb in the tuna world, and I said, "Oh, that's your problem, not mine." But yeah, so I mean, what we've done is, extensive lab testing to look at mortality of tagged fish, things like that, whether the tags hold for, how long they stay in place, things like that. And so Simon pointed out that he's done a little bit of work on incorporating both release condition and tagger experience in terms of doing some corrections for tag releases and some of the issues that may be involved with mortality right after release, and kind of estimating that initial loss of tag or mortality of tags, which I think was interesting and could be something that is explored more in depth probably, maybe in the tuna world. Misreporting, I think there's not a whole lot you can do there, except maybe release some high reward tags and hope that you get 100% return on those and do some comparisons with your regular tags. Other options are, basically, yeah, what else I want to say? You can, sometimes you can estimate the reporting rate. I know Matt and I have both kind of put that into our models, not necessarily in the tuna context, and we've been able to estimate reporting rate within simulation models, but I think more of very high quality data. So it's definitely one that's sort of iffy, whether that would actually work in the real world with messy data. In terms of representativeness, I don't know if there's a lot you can do there when you're limited by these huge ocean basins and large regions. Obviously, the ideal is to improve your spatial temporal coverage, but that's easier said than done. I do think there's some power in trying to use multiple tag types simultaneously to kind of, get information out of those different types. So essentially, you have unobserved states from your conventional tags. You could kind of fill those in a little bit if you had satellite tags and archival tags to see, what a subset of those fish are doing in terms of their actual movement, migration pathways, and use that information essentially to try to inform what the mass number of tags that you can't really see where they're moving, what they're doing. So I think there's some potential ways forward by using these multiple tag type models that integrate all these different tags. In terms of data conflicts, I think there's going to be a whole session on that, or maybe not explicitly, I can't remember. But yeah, I think there's some use of using some of these self-weighting likelihood functions. I don't know how widely they've been used for tagging data explicitly. But yeah, that's something you can kind of look into. And so these are some slides from Matt that he put together in terms of what's going on for using tagging data within tuna assessments around the world. I'm sure everyone here is probably kind of aware of all these developments, so I won't emphasize them too much. But in terms of the IATTC, we heard this morning about some of these high resolution tagging models that are being developed. That's what that figure is in the upper right. So yeah, really trying to get at issues of incomplete mixing and also estimating kind of preference functions using things like the whole slew of tags, but also electronic tags, conventional tags, but also modeling at a much finer scale so that the issue of tag mixing isn't as big of an issue. In terms of the Western Pacific, there's a couple slides that Matt provided, or a couple of figures that Matt provided. So I think it's the upper right is integrating otolith data to better estimate growth curves, using the CAPAM model to inform movement. So I think kind of the same, we heard from John Hampton earlier, I think kind of the same idea that I just mentioned of this higher resolution tagging model to better understand maybe the fine scale movement dynamics, and then trying to use that to scale up to your kind of big regions for your spatial models. What else? In terms of ICAT, I think there's been a lot of work on trying to look at mixing. So especially with the bluefin tuna models, you have two kind of populations with natal homing potentially, and trying to get at how you can model the mixing and stock composition when you have kind of mixed stock catches, things like that. What else? And then the IOTC, I mean, we had a whole, whole session that was essentially focused on IOTC yellowfin tuna the last 2 days. So I think this has been an interesting experience of using a high resolution, fine scale operating model to simulate data for yellowfin tuna and then seeing how kind of some of these models perform when you have sort of this more realistic and messy data. And then I think the main issues for the assessments are basically the same as what I just outlined, just generally for tag integrated models. The mixing across large basins is kind of the big one. Estimating reporting rate for multiple flags, fleets, collection sources, things like that. Limited spatial temporal coverage, all of these nuisance parameters, trying to estimate, realistic movement rates with limited data. And then, this is an interesting one that Matt pointed out at the end, just the lack of robust diagnostic tools to assess fit to tagging data, but also, looking at whether the movement's realistic. And so this figure is on the right, I think was showing the tagging data in the top 4 boxes and where the release and recaptures occurred, and then comparing that to the estimates within a spatial tag integrated model in terms of the movement rates. and seeing whether the tagging data and the movement estimates kind of aligned. And I think what Matt pointed out was it didn't really happen here, in that it kind of pointed out that the length composition data may have been kind of driving those movement estimates. So I think that's a useful kind of diagnostic, looking at the tagging data maybe outside the model and comparing it to how much influence it's having on some of your estimates within the model. So in terms of primary needs and future research, these are kind of discussion questions, so we don't need to linger on these, come back to them. But how do you deal with tag mixing and these unobservable states for conventional tags? And I think a really interesting one that Matt pointed out is whether, we get really mixed up in kind of tagging data and whether it's representative and whether we should be using it. And I think a more important question is that, does tagging data violate the assumptions of our tagging models more so than the other data that we're putting into the assessment? We've, based on our simulation experiment, saw that CPU data has some really strong influence, and that may be the least reliable data that we have. And so there's this trade-off of, well, do we ignore tagging data because we know that there's some assumptions being violated, but at the same time, similar things are happening in other data sets that we kind of let slip under the rug. And then there's this idea of using different parameterizations of your tagging submodel, so release condition versus recapture conditioned, Brownie versus Peterson estimates, things like that. Developing more robust diagnostics. And then, like I mentioned, integrating multiple data sources and data types to kind of help estimate movement and mortality, which is primarily why you're putting the tagging data in there in the first place. Data weighting, that's always an issue. And I really think this last one's important, is kind of disseminating results of model development when you're doing tag integrated models and spatial models. Because I have to say, as a Tuna outsider, trying to figure out what people are doing is really hard. There's a lot of gray literature out there. If you don't know where to look, it's almost impossible to find. So I have a tendency not to cite Tuna models too often because I don't know where to look, and it's hard to dig into a 150-page assessment to figure out what's happening in terms of movement estimation and tagging data. So I really think we need to work on that a lot more. It's just kind of communicating and get those results out so we can all learn from each other. And so just quickly, future directions, like this is, same points again. I think there's power and diversity using multiple tag types simultaneously within these integrated models. They are integrated models, which means you can integrate basically anything you want. So I think that's an important thing to remember, that we can start using both conventional and electronic tags and these natural tags, things like that, genetics, all of the above. And I'm really interested in these high resolution tagging model efforts. I think they're showing some real promise in terms of, estimating, especially preference functions for fish. And if you have a preference function, it really collapses the movement matrix down. You don't have to start estimating every pairwise movement between all the regions. You can kind of derive the movement and use the environmental layers and things like that to force the movement and reduce the parameterization of your spatial models. And then this last one is that I don't really know too much about, but Simon and Mark pointed out to me, is using kind of these similar idea, but high resolution simulations to look at your assumptions. So kind of simulating, I think this was using CPODIM. Probably John Hampton has some insight on the work that's been going on with this. But yeah, I think it kind of simulating your real world kind of real world dynamics at a high resolution, and using that to look at things like tag mixing and stuff like that, and whether the assumptions are realistic based on these simulation models. So just to wrap up real quick, I think Matt and I generally agreed that we shouldn't just throw away tagging data right off the bat. There's a lot of information there. It doesn't necessarily have to be integrated into your assessment to be useful. I think there's a lot of external analyses you can do to better understand the spatial dynamics present. And then, but you do have to think about, if you're going to integrate it, how do you address some of these biases that we know are there? But if you can kind of relatively well address those, then I think you can get some really useful information and needed information, primarily movement. In terms of good practices, I don't know that we have a lot of recommendations here. Again, a lot of this stuff is from my experience with more demersal species, with much smaller regional areas. So, double tagging, holding experiments, those have been useful for us. I think it's been pointed out that consistent taggers could be a useful thing when you're talking about tuna tagging. High reward tags, trying to get at, reporting rate issues is always useful. And I think just generally speaking, thinking about your experimental design before going out and just releasing a bunch of tags. So making sure that how you're tagging is going to be useful for how you're going to use the tags in the long run is definitely a useful issue or a useful approach. And then just like I mentioned, use the integrated framework. It's a powerful way of modeling. And I don't think we're necessarily using all of the data that we have. I don't think we need to use all the data, but it's worth looking at what data can be useful and thinking about that more in depth. And then again, these multi-scalar assessment approaches, I don't know that we're there yet. I think the computing limitations are still a huge problem with trying to integrate multi-scale assessments. But I do think there's a lot of information content and use for developing high resolution tagging models to help estimate movement and things like that. So yeah, that's about it. These are just discussion questions that we had, and so yeah.
Yeah, thanks, Dan. Any questions for Dan? So I got a question for you. So for the mixing period, do the tagged fish have to mix just in the box where they were released, or do they have to mix across the whole population?
I don't model tuna, so you tell me. I think the first concern is obviously within the region, but I do think, well, there's two things there. If they don't mix with the other regions, then do you have to worry about it? And then essentially you just have independent assessments. You shouldn't even be estimating movement. So.
Okay, any other questions? Yeah, Dan?
Thanks. You mentioned the needs to develop diagnostics tools for assessing feeds to tagging data. Of course, the different modeling approach is different, right? But if you're looking at multi-fancial and stock synthesis, the way they have, diagnostic looking at comparing expected and versus observed value. Do you think that's not adequate, or is there any problem with those kind of diagnostics? Or?
I don't think there's any problem with them, but I have a problem with the number of them. So with a non-spatial model, you have a set of diagnostics. You look at them, it's relatively simple to be like, here's an issue, here's not, this is working, this isn't. In the spatial models and in tag integrated models, you're starting talking about looking at releases and recaptures and residuals and recaptures by cohort, by year, by area. And so you're looking at probably hundreds of different patterns and residuals. And so for me personally, I have trouble like eyeballing that and being like, that's an issue. So I don't think there's anything wrong with them. I think the problem is how do we interpret them as humans, or develop a measure of some sort to be like, this is working or this isn't working, to diagnose those misfits and things like that.
Thanks. Any other questions? Yeah, Paul.
Thanks for the presentation, Dan. It was really good. I'm going to go back to the tag mixing one because it's a pain of our lives with tuna assessments. And this could be a very hard question. Is there any way we can develop models that don't need a tag mixing assumption? And if the tags are never mixed within the size of a region, should we still use them?
Yeah, like I said, I don't have an answer to that, but I do think the high resolution models that's why they're being developed. I think essentially is to try to, if you start modeling it by the cell as opposed to by the large region, then theoretically that cell should have a well-mixed, that should be well-mixed within the cell. So hopefully it's not as big of an issue, and then you can kind of use that and scale up to get movement rates and things like that. So you know, they're brand new, I think, more or less. I don't know if we've proven that they work or don't work. So I would say that's a good approach. We'll see how it goes.
Okay.
Yeah. So Jim Thorson has a paper where he used a very similar model to the IATTC one that's shown here. Essentially, put all of the data together into an integrated framework. Essentially, it's a spatial temporal model of tagging and movement. Estimate the movement rates by cell based on preference, advection, diffusion, taxes, things like that, and then scale up to the strata level. So essentially, it was for Pacific cod in the Bering Sea, and there are two big regions where the assessment takes place. And so it's scaled from the cell level up to the two regions and had, to estimate the movement rates between them.
Jeremy, you had a question?
Yeah, I don't know. Yeah, I don't know if this is a big question to Dan or just a general question in relation to Tuna and use of tagging, but are you concerned about tag heterogeneity and release across size or age? And if you, is it a problem? Should we be concerned about it? And what can we do about it?
I mean, I think to some extent, yes. Especially if you have age-based movement or length-based movement, then you need to have information from different length classes or age classes. Otherwise, you're going to miss what those movement patterns are. Conversely, if your tags are out long enough, you tag them small, eventually they get big and you'll have information at all the ages or sizes. But I think that depends on how long your tags last and what your fishing mortality is or your mortality rate in general. So, but I do think it's important.
Okay, thanks, Dan. We'll move on to the next presentation, but we have a lot of time for discussion for more questions for Dan later. So the next one is Rich, and my computer crashed, so I don't know anything about anything now. So he's talking about tagging of some kind.
How do I full scale? Does anybody know how you full scale a PDF in Windows? I thought it was Control L. Is that? Come on, there must be some Windows users in here. But sorry? Shit. Oh, shit, you're filming this. Sorry. Okay, well, I might have to just expand it up a bit and then do it this way. How does that look on the? Are we okay with that? Because I can just do it page by page.
Yeah, that's what we did last time, unless someone else knows the right button to push for full screen.
Oh, it's working fine. Okay, so I'm doing the comment to the keynote, and I don't think there's going to be a lot of drama here because I pretty much agree with a lot of what Dan and Matt said. That being said, what I wanted to do was dive into some of the bigger questions that they were posing and try and sort of come about, is that are there answers to some of this, or is some of it still largely unknown? So I think some questions on parameterization can be addressed. I think there's quite a bit of evidence for some of that. I think transcending is a dramatic word, but we are getting closer to moving on, if that's the right word, from conventional tagging. Genomics and e-tagging technology is really moving forward, and also the cost of being able to do it is getting much better. So I think we're getting close to moving to that magic happy land where it's not just conventional tags. We've got genomics and we've got electronic tags as well. So I think next generation technology, without being too much of the if Tony Stark worked on tag models thing, is kind of coming. I think the multi-scale approach does have a lot of promise, but also some real challenges, especially when it comes to the technological front and the computational front. But there's also some stuff that it's not going to solve from our perspective from the Tuna. And one of the things that Dan raised, I think, is really overlooked is design. Is instead of taking data sets that are just given to us and they go away and do something with that, is to have a really iterative part of that process to say, well, what do you want it to do? How much money do you have? And let's see if we can design a program from there. Because I think we've all been in that position if you've modeled tag data. Parameterization. I think this is pretty low-hanging fruit. I don't know of much evidence that says the Peterson outperforms a brownie. In pretty much any context, the brownie's definitely better for estimating M. It's also pretty much better for F and total mortality. It can be easily extended into a spatial domain. But the only thing I could really say about the Peterson above the Brownie is it's easy to explain, right? It's three variables in a nice little multiplication and division. But I think the Brownie is a more natural model for how that process works, especially when we're talking about tag models of multi-stage releases, be that age or size, where you've got repeat recaptures. I think it just fits more naturally into that space. On the spatial front, so this is work I did with Gemery over there for toothfish, and it was to justify a move away from SS3, not just because I didn't like it, we had to do say more than that, but we wanted to move to a custom model, specifically because we were not that happy about how SS3 takes the tag data. You're kind of compacting your data into the model, not having your model actually deal with the realities of the tag process. So we obviously had to give some justification for doing that. So this is a really simple 2-area model. So you've just got 2-box model. In the magenta, if it shows up that way, are the area 1 to 2 movement estimates for a bunch of different scenarios. The area 2 to area 1 estimates for a bunch of different scenarios, and the CV and the SSB. So the story being, really, if you're going to compare it to SS3, which models total recaptures and the spatial distribution of the recaptures. So it sort of splits them in two. So if you look, you get consistently better estimates for the precision if you use a spatial Brownie versus what they're doing in SS3. So I think even in that spatial context, there's pretty good evidence to say that if you've got complete spatial recapture information, the Brownie is the better way to go. Recapture only models. I think these don't get a lot of attention or the sort of affection almost that they deserve. If movement is your primary target, this can be a really useful way to make the most of your data if you're not that sure about some of the key nuisance parameters. So right, you can relax some of the assumptions for recapture only. You don't need absolute reporting rates. You only need to understand something about relative reporting and recapture rates by region. Relative recapture rates by region, your harvest rates should give you that. Relative reporting rates, that's easier to try and come up with an answer versus absolutes. So if you can do that, you can relax the assumptions of having a full mortality and movement model, but still get the movement estimates. And another point that Dan made was you can also utilize this with different types of tag data. So this was an example for striped marlin in the Western Central Pacific. So we've got the tag tracks there on the left for different release locations. We had recaptures by release location, and you can utilize some pretty good stuff called hidden Markov models, which is a really efficient way to calculate likelihoods for these kind of Markovian transfer models, integrate it all together and get movement estimates. Now, obviously the challenge is you've got to then use those in the assessment, but this kind of stuff could be integrated into a stock assessment as well. So I think recapture only models deserve a bit more attention if your focus is movement. Genomics to the rescue, of course. Like, I'll get fired if I don't say this. If you can do close kin recapture, you should go and do it. It removes all of the nuisance parameters, even the tag release one, because it's easier if they're all dead. It works really well with lethal sampling. It'll work if you release them alive. It's actually easier if you don't, which means you don't have to worry about release mortality. It can handle space and stock structure. Mixing is a different issue for this, because mixing, what you're really asking is, how does my sampling program correlate with the migration and stock structure issues? And if it does that, you can bias your answers. The flip side being, you can estimate those parameters with close kin. So if you can do it, and if you can afford it, you should just go and do it. There you go. I get to go home, back to my job, but if you can do it, you should. Gene tagging. So what does it solve? Tag loss and reporting rates, but it doesn't obviously solve release mortality. How are you going to do that? That's very difficult to solve. Does it work in the big tropical species context though? We've done it for SBT. That context was quite specific, so it was easy for us to release and look for recaptures in fish because we're doing it at the same localized spatial location. We can get clean samples with very little contamination, and they're not quite as big as some of those big tuna species. So does it work at this point with the tech that you need in terms of genetic sampling with a minimization for contamination? And also, how much does it cost to process all those samples? It's not clear that that'll work for the big tropical tuna species yet, but it could. eDNA, not really. I don't see that one. If your event space of possibilities, you get a bucket and you say you've got a skipjack. Hypothesis 1, there was a skipjack there before. Hypothesis 2, it got eaten by a massive predator over there with this slow digestive system that moved over here, took a shit, and there you have your eDNA. That's a pretty wide event space, and I'm not sure how you model that. So I'm not convinced that eDNA is going to do as much good yet. Promise and pitfalls of spatial temporal scales. There's a real appeal to this, right? If you move down the spatial temporal scales where you can actually accommodate what you're really worried about with mixing, you've got those finer spatial temporal scales, and then you can rescale this back up to the assessment. So I think if you're modeling the tag dynamics at scale, where the processes really matter, and then you rescale it to your model, that's taking out one of the really difficult sort of problems. The question is how feasible that's going to be in an integrated sense. How long does it take for one run of multifan, Claudio? Okay, so that's going to be a challenge if you layer onto that a very complicated model, either a really detailed stochastic differential equation, or if you go the full partial differential equation approach, it's something called the Fokker-Planck equation that models the time evolution of the probability of capturing something. They are way more expensive than even our most complicated models that we use today. So they're going to be a real challenge to layer underneath. I think that's a sequential approach for things like that, where you do that analysis first and then use that data. You try and impose that distribution that you've estimated, but not do it concurrently. I don't think we're there technologically in terms of the computational cost to do that. It also doesn't solve the design problem, right? It doesn't matter how good your spatial resolution is. If you've got a badly designed tagging program, just modeling the dynamics at scale isn't going to fix that. If you tagged all your animals on the border of a region, you're going to get a biased up estimate of how much moves. So it doesn't solve the design issue. How many people have tagged data have been through this story? You get the gift of the data. You don't really have any understanding of how it was collected. It wasn't that clear what they wanted from it in the first place. And then you go back and you try and do some modeling with it, and people are like, that's not what we signed up for. Why can't we estimate all this really cool stuff? And I think given the expense of large-scale oceanographic tagging programs, it's about time we got a better way of designing these programs for the objectives they're supposed to meet. Often, I think we fall really short of the expectations of the people that are financed or approve this kind of thing, and they say, why can't we estimate those things? Because I don't think we had the part in the design. So the IATTC one is probably a case in point. So below there, I think, Dan, this is your graph. Thank you, Dan. So the green dots are the releases, and the heat map is the recaptures. I think that's right, isn't it, Dan? I want to see a nod. Fantastic. If you were asked to design a program that was going to give you abundance, mortality, and movement for the Indian Ocean scale, you probably wouldn't come up with that. That's more a design of necessity than function, right? Is that kind of measures, basically, it's a product of where they could release fish. But if it doesn't work, is it worth all of that investment? Could we do better? Like, is it better to design a program, talk to the people and say, this is what you'd have to do if you want it to do this, and then say it's too expensive, then go and spend that money and not being able to do it? I think that's the question we need to look at. So I don't think that matters whether it's conventional or genomic or electronic. Design, you should do the work first. It's not worth wasting that money if you wind up with something that doesn't really work. So summing up, conventional tags, they've had a bit of a rough tuna ride. They showed a lot of promise, but they've also got a lot of problems. I do agree we just shouldn't be throwing them out. Standardizing the CPUE of various esoteric different fisheries is not necessarily going to be a game changer to give us abundance information that we want, I think. There's a lot of advances in new tech where it comes from electronic tags or genomic that I think is going to show some real promise into the future, where I think some of these tech cost thresholds are going to be workable at scale on an ocean basin. How do we dovetail all of this within the integrated assessment ethos? Some of those downscaled models that are really complex, I think that's going to be a challenge to put them into integrated assessments. And lastly, I think we need to do a better job of designing these programs to deliver what people want them to. Okay, thanks very much.
Thanks, Rich. Do we have any questions? Carolina?
Hey, Rich, so we're talking about design. How will you go about to design a study, such as one for the Indian Ocean, for example?
I mean, you can go the full simulation evaluation route. That's quite expensive and difficult. Some of the stuff we've been doing for the close-kin was more around getting a handle on what sample sizes you think you're going to need to deliver the precision of the things that you want. So there's some nice statistical tools that have been developed by Mark Bravington and a few of us that can pretty rapidly give you an estimate of the uncertainty in the things you're interested about, given the specifics of your sampling program. So sample here, this many fish of this size group over this time frame. So I think we've got some statistical tools to do it. And the nice thing is, if you're using most of the tagging models, they generally have either a binomial or a multinomial or some type of distribution like those two. They fit really neatly into those kind of design processes. So we've done some stuff for the IATC along this front, fully spatial close kin. They run pretty quickly, and it's not about bias and things like that as much as initially about precision and how many samples you're likely to need and where. So we've definitely got the tools to do it, and you can just switch it for tag models. So this work was done using 2 spatial tag models, same general principles, looking for the precision of the estimates given the sampling specifics, not necessarily a full error bias profile.
Okay, any other questions? I got a follow up on that, not necessarily for Rich, but for other people. Given the practical issues in tagging tunas, particularly tropical tunas, is it ever going to be possible to develop a tagging design that's actually going to work?
I think it's that was the right sign.
I actually
How
don't.
desperate are they to get an answer?
I don't think that's the sign, because the problem is being able, well, maybe there is if we collaborate with industry and are able to use their echo sounder buoys to catch the fish and stuff like that. But we do have a serious problem in trying to find fish and tag them, right?
Yeah.
And so it may be possible, but it may need a lot of collaboration with industry and maybe improved technology and things like that as well.
Yeah, and the genetics has been proven to not necessarily doing the sequencing at that size of numbers, but getting clean samples has proven a real challenge. Like, we developed a tool to do it for SBT that works in that controlled environment where you get a sample and it's clean. The worst thing you can get is cross-contamination, because then everything looks related to everything else and you're pretty much done. But getting that out on a longliner or sampling inside of a tuna market, to get a biopsy, whatever. All of those are going to be real challenges at scale that I think you're right, are not necessarily cost but tech related.
Actually, what I was talking about was conventional tagging rather than close kin. So my next question is, what's the cost difference between a conventional tagging study and a close kin now, in an order of magnitude type thing, rather than, are they close?
They were close enough for CCSBT to say we're happy to switch and finance it. I mean, the close kin's a different one because you don't need to go out and you just sample fish that have already been caught. That's a big advantage. It works easier if they're already dead.
So I think we have a question online, do we? Yeah, John? Yeah, okay. John Hampton, do you want to ask a question?
Yeah, no, Mark, it was kind of responding to your first question that you raised about, can we ever design a tagging program that will be effective? I mean, in a sense, the design is the easy part. The implementation of that design is the hard part. And of course, with tropical tunas, we're very limited in needing to catch fish in large numbers for a conventional tagging experiment in a condition that they can be released in order to minimize tagging mortality. That usually means you have to use pole and line gear, which severely limits the spatial range over which you can do the tagging, because you need to have access to bait in order to employ that methodology for tropical tunas. So, that's the major limitation. I mean, like Rich and I think many others, we've all been kind of at least somewhat seduced by the promise of genomics and close kin mark recapture as being a potential solution to this stuff, and we have some work underway to trial that for South Pacific albacore. So we'll see how that pans out.
Okay, thanks, John. So I think we'll move on to the discussion period, but obviously you can ask Rich some more questions during that, and also Dan, and any other comments or points. So does anyone have a comment or a question that they'd like to ask? No? Oh yeah, Aaron.
Yeah, thanks. I just was going to ask a question mostly because I want to hear Rich talk about eDNA some more. But it does have to do with that. I was just curious, and this is just thinking outside the box. You know, typically that's been used for kind of presence absence, right? And there has been some recent work in the Eastern Pacific where some eDNA work by Ole Shelton and others showed that for Pacific hake, it actually matched the survey, the acoustic survey estimate of abundance. So they actually scaled it up, and it was a time series, or not a time series, but a particular year where it matched abundance fairly well. So there's a little bit of promise there. I know it's very much in its infancy, but if in fact that did work, I was wondering if there is a utility there, given it sampling water. I mean, you can imagine pairing that with VMS and taking samples from a hull of a boat. If that would be useful, even as insofar as confirming or refuting, you know, our regional scaling, you know, using our CPUE for regional scaling or some sort of other aspect like that, that's not going to solve, you know, movement perhaps, but can be used in other ways. So just a comment.
Okay, does anyone want to reply to that? Yeah, Rich.
So I can slightly walk back some of my slightly flippant comments, but I'll stick by most of them. I think there's two bits. There's the way that eDNA could be used as exists now, which is more in that sense that might have some utility, but it's the additional hypotheses of how it could be there that is a real challenge, because you kind of focus in on the question you could answer. You have to step back and think, what are the other possible questions or the other possible hypotheses for how I ended up with that in my bucket? That's a challenge. I think the second one is technological, which may get solved, which is to get cellular level eDNA, where instead of just saying that's from a species, you're saying that's from a fish. And I might have seen that fish before, and I might see it again. And I think that's a different level of really, they're not there yet at all, doing that with eDNA. And they do make a lot of promises, and some of them don't come true. But if that one did, I'm willing to see that one as where it's really starting to be a potential game changer. But then you've got the challenge of sampling effort. And yeah, but I still think the technology is not there yet, but I think watch this space, I guess.
Any other comments on that particular topic? I got a comment. I'm pretty sure that no one here knows anything about this, so we're probably not going to solve these problems. But if you sampled in everything, you know, just randomly sampled over the ocean, and you measured the eDNA, and your eDNA thing gave you a relative abundance of eDNA in that sample, and then you did a spatial temporal model of that, and then modeled it, and then now you've got a good relative index of abundance. Why would that be any different than, say, I mean, you wouldn't have length frequencies and stuff like that, but at least it would have some form of index of abundance. Wouldn't that be similar to what we're doing with CPUE and other things? Yeah, Rich.
I think it could be, but again, like with CPUE, there's a lot of things to standardize how that DNA was shed. Mm-hmm. And without being in situ or doing a lot of experimentation, it's quite hard to know that. Mm-hmm. If they're in schooling species and they're in quite turbid conditions and stuff, you could imagine they're shedding quite a lot. Big animals that are fishing on, you know, swimming on their own, not encountering a lot of solid objects and things, might shed quite a lot less. So there's the lure, and then there's the reality of what else you might need to know to make it work. Because in principle, it could and almost should. But I think there's some real challenges to how to standardize the density of DNA that you actually observe.
Yeah, I'm sure environmental factors and stuff will make a difference too. And anyway, something for the future maybe. I'm sure someone's going to look into it. Okay, any questions? Yeah, Nicola.
Yeah, thanks, Mark. This is for the group, but it's particularly aimed at those that have more experience with the genetic tagging techniques or close kin, and its ability to perhaps address, if it can, age-specific movement, because that's one of the, I guess, the frustrations with conventional tagging data, especially based off of fisheries-dependent tagging and recapture platforms, is you tag often one size or age class, and you may or may not see a representative recapture history. So you're often assuming a single movement pattern for an entire population that we know from life history that they're probably not all moving the same way. So some of these new techniques, do you see the potential to try to do better at that, or are we still kind of stuck in that aspect?
Rich.
Talking a lot, hopefully stop talking. I think splitting it into juveniles and adults is a good way to delineate the two challenges there. With the adults and the close kin, yes, you should be able to, in principle, get, if you've got estimates of size and or age of the adults that you're sampling, you should be able to get estimates of the movement. How much they're going to differ in terms of their movement patterns if they're old enough to be parents, there might not be a lot of change there. But you could, if it was there, you could in principle find it. The juveniles one is a challenge because close kin is all about adults, but it's not all about adults. But in a spatial sense, if you thought about, the stuff in the Indian Ocean, we didn't go there because the amount of Greek it takes to just explain the probabilities when juveniles and adults move is not very nice. But it is in principle possible that if you're sampling in each area and you're sampling juveniles and adults, looking for the parent offsprings and the half sibling pairs, that there's information on the juvenile movement as well, because they have a probability of being sampled in a particular space when they used to be somewhere else. And it gets all bundled up into the probabilities of, is that animal related to this animal? So in principle, you can get the juvenile aspects as well, but technically it's really complicated, but it's in principally doable.
Okay, thanks, Rich. Anyone with a follow-up question on that? And I'd like to have a follow-up question. So close kin mark recapture focuses mainly on the adults in terms of natural mortality and absolute abundance and things like that. With our tropical tuna fisheries, we have a large amount of the catch on juveniles, right, from the purseine fisheries. So if we just used close kin mark recapture, would we be okay, given we have maybe link frequencies or something for the other sized fish? Or would we also need to combine it with a conventional tagging study just for the juveniles, which is something that we can kind of do? It's not the greatest, but at least it's more practical than adults at this stage.
Yeah, sort of CCSBT made that call. It said that close kin won't be enough, especially given the sort of slow maturation of the southern bluefin. Is that we need something else for the juveniles. That's not going to be enough. So it's in the assessment, it's all combined together, and in the management procedure, they're all combined together. So I think it's hard to imagine. Mark's not going to like me saying this, but I don't think you can just do all of it with close kin. Is what I agree with you, especially for the stuff that takes a little bit more time to mature. There's just too much missing, from year class strength and other things. So,
but would link frequency data or something like that and catch be enough, or would you have to have CPUE for the juveniles or an index of abundance?
I mean, you're still going to have that anchoring adult abundance. So you just got to hope you've got enough information to get the juveniles there in terms of there being enough to give you the number of adults you need to represent the close kin data. But it's how much uncertainty do you wind up with along the way? Because you've got the adult abundance with a particular precision, but You know, if you looked at the estimates when you do close kin only, and you look at the estimates of R0 and things like that, they're pretty damn wide, because it's so far back in the chain that it's quite uncertain. You can do it. I think it's still better to have as much data, decent data as you can on the life stages.
So it might be useful to have a simulation analysis for Tuna to work out where the close kin is actually going to do what we want. John's got a question on?
No, Matt. No.
Who? Matt. Matt.
Yeah, I was just going to make a quick comment about the recapture condition that Rich talked about. My understanding was that SS used the recapture condition in its likelihood, and that was why you had the less precise estimates, because I've done some simulations that compared the recapture to the release condition, and the recapture condition generally had a higher uncertainty than the release condition. And I guess I'd also like to make the point that the math on the recapture condition is reliant on the assumptions that those nuisance parameters are constant temporally and spatially. And if you have spatially varying reporting rates, that would be caused by reporting rates that are very different between ports of recovery or something like that, then this can really mess with your estimates of movement. So I'm not sure if there's a way to overcome that assumption violation, and if it's worth trying to do that with the Rich said something about the relative reporting rate, and I'm not sure that the maths work out correctly, because they don't cancel out in the likelihood, but I don't know. That's getting very technical, but.
So we're probably talking about two things though. So when I said recapture only, it really is removing all of the releases. So slightly different to what SS3 does, where it does have the releases and recaptures, and it conditions on overall number of recaptures and the spatial distribution of those recaptures. So that was the bit of SS3 I was talking about. With the Stripe Marlin one, it uses just recapture only. So it doesn't use the releases, it just models, you know, conditioned on the fact it was recaptured, you only use those ones. And you can have time varying differences if you know what they are in the reporting rate and the relative recapture probability. If you don't know them and you're not including them, they can definitely bias the answers. But the maths doesn't need to have constant rates. You can input those. That's what we did for the Stripe Marlin, is to put the relative probability of recapture being time dependent. And I think the reporting rates were constant by area. But if you have the answers and at least you have estimates to try and put those time varying estimates in, they don't have to be absolute, but the math still works.
Thanks. Matt, do you want to follow up on that, or that's okay?
No, it's all good.
No. Okay, any other questions? Okay, I got a question. Oh, Carolina, go. Yeah.
For Rich as well, or any other person who knows a bit about this. So there's some literature about aging fish with DNA methylation and things like that. Are you guys exploring any of that in your labs? Can you comment on that?
Yes, I mean, it's probably most advanced for SBT, but I think also for Indian Ocean yellowfin as well, in terms of calibrate. And that's the key bit, is to be able to calibrate it. So you've got the samples that you ideally want with paired age estimates to get a calibration for how the methylation of the genome proceeds as it gets older. But it looks pretty good. So the SBT is probably most advanced in that regard, but yeah, looking into that for Indian Ocean yellowfin. And I think for some, I'm not sure, I can't speak for Western Central and some other ones, but it's sort of been assumed that you'll do that if you're going to invest all the money with a close kin program, is to just include that genetic aging aspect.
Okay, any other questions? No, I got a question for Dan. You talked a little bit about high rewards to estimate reporting rates and things like that. If you estimate reporting rates, particularly spatial reporting rates, not necessarily over time, assuming they're constant over time, How much information have you got left in your tagging data once you've done that?
I have no idea. Yeah, I mean, I think that's why if you have kind of the high reward tagging data, you can add an objective function component for fitting the high reward tags explicitly. And I think you can use that to estimate the reporting rate without assuming, you can actually estimate it explicitly with its own objective function component. And I think that's a useful approach. But I don't know if I have an answer to your question explicitly.
Yeah, it was kind of a bit of a loaded question, because I did an analysis. Someone did some work and I was a bit worried about it. So I did analysis and I can't remember exactly the details, but it was basically on high rewards tags. And when I did the analysis, and I can't remember the assumptions and stuff, but the analysis ended up just being an analysis of high reward tags. All the other information from the other tags would basically eliminate it. And so you might as well have not done them and just did high reward tags and that was it. But I can't remember if I had time varying reporting rates or what it was, but yeah. But I think that would be a useful thing to look at in your studies, and it would be easy to do with, likelihood profiles and things like that on the different tag types. Yeah. But I know that at least the Western Central Pacific are estimating a lot of reporting rates, so maybe their analyses would give us some insight. Okay, any other questions on? Yeah, Dan?
Yeah, so I'm pretty naive with the close-kin market capture stuff, and I was kind of interested, when you do your, you get your absolute abundance estimate, that is for the entire population across all regions. It's not, like region specific, unless you have multiple populations. Is that right?
How do you turn this on? Just talk at it. Is it on? That's good. What was the question? Oh, yeah, okay, region specific. So by the time I got it working, I forgot. Yeah, so I mean, in the simple case, if you've got one spawning population, it's not region specific, it's just for that whole region. If you do have, it's probably worth splitting it into, do you have spatial structure, but not different stocks? Then it is region specific, and you'd want to account for the migratory and relative density of those regions and your probabilities of, is something related to something else in a different region? And then if the stock structure in there, again, that's the next level of complexity that you've got to build into the probabilities for, is this thing related to that thing in this particular way? So yes, if it's there, and you can bias it if you don't really account for it.
Yeah, because I mean, I'm just interested in this, there's multiple regions in all of these tuna models essentially. And so I guess the question is, if you can get a good absolute abundance estimate for the entire population, do we care about what kind of the regional population, the regional abundance estimates or biomass estimates are? Do we worry about the local depletion kind of situation, or does it not really matter? And so I think that's an interesting one for the close kin, whether we care about that or not.
Okay, any other questions? No. I guess I'll ask, yeah, Nicola, you got a question?
I was just going to comment on what Dan was saying. I mean, I guess it depends on your management framework whether or not you care about, like, if you're able to get a really good estimate of the total stock, like, are they basing any management decisions off of sub partitions of that total stock, and that probably guides the approach that you need to take.
Yeah, Dan?
I guess my follow-up is more, should we be worried about it? If the managers aren't worried about it, that's one thing, but as scientists, should we be worried about if the depletion in one area is much higher than a different area? So I think there's an interesting discussion in there of, managers sometimes worry about different things, and it's not necessarily the science or the biology that they're worried about.
Yeah. Okay. I got a question. We keep on going back to close-kin market recapture, but I guess it's kind of a trendy thing. So my understanding is close-kin microcapture can actually estimate the stock structure, right? And we've had a lot of difficulties estimating stock structure. And I was wondering what sort of movement in and out of the stock does it detect? I mean, you know, for assessment purposes, a bit of movement in and out is not really that important, right? But with the close kin stuff, you know, how much movement in and out can it detect, and whether that relates to, you know, the ability to do stock assessments?
Yeah, I mean, it's that point of what movement is important. And I think if it's really low, like let's pick a magic number like 5%, because that's significance everywhere. But if it's less than that, you'd probably have to sample quite hard to find it, but do you care? So I think you're probably saved by the fact that if it was really quite high, it wouldn't be so onerous in terms of the sampling level. You'd have to see it. Like you can see it qualitatively straight away. Just if you find two juveniles from different areas, you know, pretty sure that they haven't moved. They were born in completely different parts of the ocean, yet they both happen to share a parent. You're pretty damn sure that those parents are moving between those areas, and vice versa. If you never see that, you're pretty sure that they're not moving. The quantification comes from the modeling aspect of it. Yeah, that's true. But I think if it's really low, you'd have to sample hard. But if it's really low, do you care?
Okay, any other questions on any aspect of tagging or using tagging data in the model? Are there any RFMOs that are actually actively doing tagging programs on a large scale? Something that would be, say, consistent of estimating, you know, total abundance for that particular stock? Yeah.
Sorry. So CCSVT is doing gene tagging for 2-year-old abundance. It's not as large scale as you would need it to be to do it for skipjack, but that's just a question of the relative scale. But it's still genotyping tens of thousands of fish every year and looking for the same fish between one year and the next. So yeah, that's been running since 2018.
Okay. What about conventional tags? Anyone in the tropical tuna world doing that? I mean, is SPC's tagging program fairly substantial, or is it sort of more of a ongoing sort of limited tagging process? Yeah. Yeah.
Hi, Japan have been conducting skipjack tagging research, and we have a historical data maybe from 1916 to current, and also we try to examine the relationship among regions, and also we are now doing major tagging research in tropical areas and also temperate areas. Yeah, that's just sharing the information.
Yeah, thanks. So what about the SPC group? What's their current tagging program look like?
I'm not that well qualified to speak, but I know there's skipjack tagging cruises that was done last year. It was a tagging cruise concentrating on skipjack. There are a few other tags done of, I think it might have been 1 or 2 big eyes and a few yellowfins, but the focus was on skipjack. But yeah, other people are much better placed to comment than me on that, but there is some happening.
Yeah, Paul, do you want to update? And also tell us what your future plans are too, would be good too.
I'm not sure I can know what the future is, but for now we tend to have different cruise types. We tend to alternate between cruises that, and John Hampton will be able to pick me up if I'm wrong, we tend to focus on skipjack in the Western Pacific, so around the Solomons, PNG area. We tag a lot of skipjack there, you know, moving, you know, a little bit latitudinally in that range. different stations. And as John mentioned before, it's using pole and line vessels, and you have to be able to get bait and things like that. So it's a bit restricted in how far it can range to tag the numbers of fish that we need to do, which is typically 10,000s or more a trip. And then other tagging cruises move into more towards the Central Pacific, and that's where we might target some floating objects and things like that to try and tag a yellowfin big eye. Again, they're often smaller fish, and that's an issue, I guess, that Nicola alluded to before. The size range of the yellowfin and big eye, we don't tag a lot of big fish, and so it's a little bit biased in terms of what we're representing with that program. But the tag program it's part of the ongoing funding that we get from the WCPFC as part of our service provision. It's about a $700,000 a year allocation that has been ongoing for quite a number of years. And as far as I can tell, there's no indication that that's going to slow down. So we do expect to continue tagging on roughly the same scale as we have been for the last probably decade or so. But yeah, it is, we're starting to perhaps look at alternative ways of improving the way the design of it is, but it is, as John said, it's very difficult to spread it out across space and life stages as what we might want to do in an ideal program. So and that creates challenges for the assessment, obviously, and we've spoken about a lot of those. But yeah, it's still ongoing. Yep.
Okay, any other comments? No. Okay, Dan, how many operational integrated tagging stock assessments are there in terms of ones that actually provide management advice?
I don't know, talk to Andre. I mean, outside of Tuna, I would say there's probably like, ballpark of 10 to 20, but I don't know off the top of my head. I'm not an encyclopedia.
So how many do you know of, like in the US? I mean, is your sablefish being used for assessments? Is the yellowtail being used for assessments?
No. The sablefish one is just being developed now by Craig. He's working on it as we speak, I'm sure very hard. So that one, and the plant, it's one of those things where getting it into the management realm, there'll probably be parallel assessments, and we'll kind of compare and introduce people to it very slowly. And the idea of having that sort of complexity in the model and whether it gets accepted is a whole different ball game. In terms of NOAA-wide, I mean, I don't know if Aaron, you have anything off the top of your head, but tag integrated, I don't think there's any.
And what are the models you're developing? Are they your own coded ones, or are they using stock synthesis?
The ones I've done were just, my own coded ones, and they got thrown out almost immediately.
Okay. Yeah. I guess at some stage maybe we can get some feedback from the online people about whether stock synthesis is actually being used as a tag integrated model for management in any species, apart from tuna. Yeah. Okay, any other questions on tagging analysis, tagging models, tagging data, anything? Okay, looks like we're a bit tired for the day, so I guess we'll probably end it there, and we'll be back tomorrow morning at 8:30, I think that would be it. And we're talking about Persane species comp and size data, recruitment, CPUE, natural mortality, and weight length, maturity, conversion factors. And the pub for the day is what?
It's the Haydai in Haydai at 264A Cuba Street.
Okay. And what time is everyone turning up?
That I hadn't planned.
When they're thirsty.
When they're thirsty, yeah.
Okay. Right now then, yeah. Okay, thanks everybody. We'll see you tomorrow morning.